A new autofocus technique developed at Caltech could eliminate the time-consuming task of focusing a variety of microscopes used in labs worldwide. Dubbed Digital Defocus Aberration Interference, or DAbI, the technique uses a standard microscope imaging setup and sample placement. Researchers light two LEDs independently to illuminate the sample from slightly different angles. A camera captures an image during each illumination, and each image is distorted in its own way. DAbI doesn't work directly with these raw images. Instead, it makes a Fourier transform of each image, a complicated mathematical operation that maps frequencies across a domain. When the two transformed images are added together, there's an overlapping region where the two signals interfere, creating a fringe pattern. The spacing and shape of that fringe pattern indicates how far out of focus the sample is. The system can calculate exactly how far the sample is off the focal plane and then make the proper adjustment to create a focused image. The whole process takes less than 1 second and produces sharper images than current state-of-the-art computational focusing methods. DAbI has the potential to save researchers and laboratories significant time at every stage of their work.